All right, good afternoon, guys. My name is Tom Chadwick, and today I'm going to be presenting rabbit fever, also known as tularemia. And so this is a disease that is going to be affecting mammals, and as well from the mammals themselves, small rodents, human beings can be infected. There is no human-to-human transmission of this disease. So first things first, the bacteria itself, Francisella tularensis. So this is a gram-negative coccobacillus. It's aerobic. It doesn't form spores. It's non-motile. And so the taxonomic classification, domain bacteria, phylum proteobacteria, the class Gamoproteobacteria, order Thylotrichomes, family Francisolaceae, genus Francisella, and then specifically the species F. tularensis. And so on the right-hand side, we see some depictions of Coccobacillus, which is essentially going to be not quite cocci, not quite bacilli either. So it's a little bit of a morph of the two. And then down on the bottom, there's another depiction of what that looks like. And so now we're going to talk about the strains and the family itself. Once again, the family is Francisolaceae, and they are noted by their convex shape, their white color. You see their colonies on the left-hand side are resembling, mucoid. And then they are generally isolated and grown on a specific agar, cysteine agar, and that's how they can survive. So the species itself, F. tularensis, there's two subspecies of this, the first being F. tularensis SSP type A, and that is the most virulent of the two, and that is the one that we associate with rabbit fever. The second subspecies is F. tularensis SSP holarctica type B, and that one can also be infectious. However, we do not associate it with rabbit fever itself. All right, so the pathogenesis, right? So what we're going to have is this bacteria. It's going to enter the body through its vectors, and then it's going to proliferate in macrophages, dendritic cells, neutrophils, hepatocytes, endothelial cells, type 2 alveolar epithelium in the lungs, right? And so the main route for entrance for infection of the cells is phagocytosis. However, the mechanisms that it takes to take or to infect cells that are not phagocytic, there needs to be further research done on that. We do not know how that quite works, but when it comes to the main route of entrance for infection of these of rabbit fever for this disease, it's going to be through phagocytosis. And so some of the receptors that it can cling to is the mannose receptor in macrophages, as well as complement receptors in macrophages, neutrophils, and dendritic cells. And so after it is taken into one of these cells, it resides as a phagosome, and then it will mature into a phagolysosome. It'll actually acquire early markers of some of these endosomes, some of these vacuole-assisting cells, and that can kind of help it live and survive and mature inside of the cell once it enters. And then once it's matured, it will physically disrupt the phagosomal membrane, and it will enter the cytosol, will replicate like crazy. And so it actually has some functions that can help it evade the internal immunity. And so as we know, bactericidal functions of phagosomes, basically, they can produce reactive oxygen species, and they use NADPH oxidase as the enzyme to convert oxygen to superoxide and hydrogen peroxide. But our species here, F. tularensis, is able to disrupt this reactive oxygen species from generating. And what it does is it alters steps in NADPH oxidase activation, and it inhibits some of the assembly. So here's a good depiction kind of what happens here. You're going to see this phagosome, and it's going to be entering into the phagocyte. And then once it's there, it's going to interact with the early endosomes and late endosomes. And once it matures, it's going to break out of the phagosome, enter the cytosol of the cell, and then just replicate. And then it's just going to kill. All right, immune response. So intracellular immune response, generally, you're going to have innate immunity. It's going to recognize the receptor patterns, the PPRSs. And these are on the cell surface or on the endosomes, and these are those toll-like receptors and those nod-like receptors. And they're going to recognize those pathogen-associated molecular patterns, PAMPs, DNA, ATP, all those good stuff. And this is going to trigger pro-inflammatory response. It'll trigger cells known as inflammasomes. And this will lead to cell death via pyropoptosis. It's a fun word. So a brief history. It was first isolated in the USA, and this was after an earthquake in San Francisco in 1906. A gentleman by the name of G.W. McCoy, he was the head of the U.S. Public Health Service Plague Laboratory. He went down to San Francisco to test some rodents to see if they would carry plague, because plague is associated with generally, dilapidated conditions, high volume of people. This is really important. And so he went down there and he started doing research and conducting studies on some rodents, and he found this bacteria. So it's named after Tulare County, which is a site of discovery in California. And another gentleman by the name of E. Francis, he was conducting research and tests specifically on this bacteria in 1919. And he found some research in Japan showing symptoms of people who may have had contact with this bacteria. And so an individual by the name of Hakiro Hara actually agreed to have the live bacteria placed on their hand. And then it led directly to disease, which E. Francis then isolated and found that it was our bacteria. And so that's the first case of it being shown in human beings. Some of the simple epidemiology here, mostly it's endemic in northern hemispheres. In America, it is in all states except Hawaii. And between the 70s and 2015, there are roughly about 200 cases in the US per year. Most of these cases are going to be reported in the summertime. And there's also wide distribution in the Eurasian continent. There's high prevalence in what was the Soviet Union and some Nordic countries. The reason for the prevalence in these places is kind of unknown, but it is interesting to say that in what was the former Soviet Union, you have areas of high population density following a post-war time when there's a lot of destruction and you don't have great infrastructure. I mean, this could be a direct way for rodents to contaminate water sources, for food to be contaminated. And so you do see something like that. In the 90s in Kosovo, there was a high rate of this disease after some of these cities were just torn up by war. And again, it's because you're destroying the infrastructure, and you're just opening yourself up to being contaminated with disease. And so from the CDC, we have a little statistic of the United States of these reported cases in 2018. And the association with these with arthropods, which are blood-sucking organisms which are associated with this disease, you may see some instances of that in the center of the country. With that, though, you would expect to see more in the north of the country, right, as we've seen with some of these other projects. But the Midwest, and specifically the middle of the country, you might see this disease more often than not due to the fact that there is high agriculture and high farming in these areas. And so some of the yearly reported cases, again, that average 200 cases per year in the United States between 1950 and 2018. From the 70s up to 2015 is when we kind of see that average. Beforehand, the cases were very, very high in the 50s. Not sure why that is. It could be that people were just diagnosing certain things, maybe not confirming that it was in fact tularemia, but and so it happens more in males than it does in females. We see this distribution here of cases, as well as the age groups. It's usually happening to middle-aged and young children, but specifically males, right? And so I think that's associated generally with just how people, how males specifically on average may go out more, or maybe more associated with work that involves agriculture and farming, where they're more susceptible to getting this disease and facing this bacteria. And then it mostly happens in the summertime, May, June, July. That's probably just because people are more out and about. And if you have ticks and fleas and horseflies coming around, summer is probably the best time for them. Most people are around, so that's probably why we see that. So again, the vectors and reservoirs, arthropods, like I said, we're going to have ticks and deer flies. It can also be skin contact with infected animals, drinking contaminated water, or inhaling contaminated aerosols. So generally this is associated with hunters hunting rabbits, actually. And so that's where we get the word rabbit fever. But yeah, so hunters are dressing the animals after they kill them. And so you're going to see that contact. And then the reservoirs, there's some data that says it's potentially in mammals, like small rodents. But there's some data that also says it's most likely not going to be in those rodents because they would die too quickly. So it's most likely in non-living objects, can survive in mud and water for a pretty long time. So the signs and symptoms, this disease actually expresses itself in a couple of different ways. The first being ulcer glandular. This is the most common, usually after the bite of one of those insects we were talking about. And it's associated with skin ulcers and swelling of regional lymph glands in the armpit and the groin. And then the glandular itself is another way that this disease shows itself. And there's no ulcers specifically, but you will also get swelling of lymph glands, armpit and groin. Ocular glandular is actually going to be contact through the eye. So after you're dressing something, hunting, in contact with it, you're going to touch your eye, and that's going to lead to conjunctivitis. Pink eye, which is essentially just inflammation of the conjunctiva, which is that tissue that lines the front of the eye and the eyelids. Oropharyngeal, that's going to be associated with eating or drinking contaminated items, and you're going to see sore throat, mouth ulcers, tonsillitis, and swelling of lymph glands in the neck. It should be noted that all of these are accompanied by a high fever of about 104. It doesn't necessarily have to be that high, but it can get to that point, which is extremely, extremely detrimental. And then the most serious and worst form of this disease is the pneumotic form. That's going to be cough, chest pain, difficulty breathing in dust and aerosols is kind of how you would get contaminated and feel these effects. And this can happen also when other forms are not treated. This bacteria is going to start traveling through the blood. It enters the alveoli of the lungs, and that's how you're going to get your pneumonia. And so the most serious signs and symptoms are, again, going to be these ulcers that you'll see on the skin there. And so some diagnosis and treatment. It is a rare disease, so it's difficult to diagnose. And the symptoms are kind of common, so you just need blood tests and cultures to confirm whether or not it is the bacteria. Generally, it's treated with antibiotics, which are streptomycin, gentamicin, doxycycline, and ciprofloxacin. It takes about 10 to 21 days for the treatment, and then most people completely recover. The mortality rate is about 1 to 2% with antibiotics. Without antibiotics, maybe about 5 to 10%, which is pretty high. But so that is all I have for you guys today. Thank you very much. Enjoy the rest of your week.